So it is my pleasure to introduce Dr. Stephen Jenkins. He is a doctor of internal medicine at the University of Utah Hospital. He's been on the board of trustees for several years and is a member of CRF's Scientific Review Board. He is also a co-chair with me this year for the Day of Hope, and most importantly, the father of 3 beautiful kids who I'm sure you'll see running around here, 2 of whom have cystinosis. So welcome, Stephen.
Okay, it's a good-looking bunch. So, going to talk about the basics of cystinosis. So we call this the building blocks. This is also commonly called the Lego talk now because we talk a lot about Legos. This week, because the conference has moved to San Diego, we finally made it to the Lego Mecca, just north of here. So there's Sam and Lars, my boys, at the entrance, and it's no Disneyland, but we had a good time, and we geeked out and got to see all their favorite Legos. So, all right, so why am I up here talking today? So this is my son Samuel when he was 12 months old, and that's when he was diagnosed with cystinosis. So in a very familiar story, he had stopped growing, he was throwing up all the time, constantly dehydrated, just an extreme amount of thirst, and just not doing well. And it's a story now that we've all heard so many times that any of you could make this diagnosis if you saw a kid with these symptoms, right? But our pediatrician was flummoxed. It took a lot of doctor visits before they figured it out. And to be honest, the first person who I think proposed that it could be cystinosis was actually my grandma, because I have 2 cousins with cystinosis, Amy and Emily Shaw, this is a picture of Emily. She was 4 years older than me. So I grew up, I'd heard of cystinosis. I knew that my cousins had this condition that made them short. I knew that they had kidney problems and they had kidney transplants, and that they really liked salt. That's about all I knew about it, but I also remember my parents telling me, yeah, we don't know how long they're gonna live. And that was when I was like a young kid, they said, maybe late teens, we don't know. But this is Emily when she was 40. She actually did pass away a couple years ago. So when my grandma saw Samuel at a family reunion, it was, I think it was Memorial Day weekend, she was watching him drink water, and just like the look on his face when he was drinking that water, she said, I think he might have what Amy and Emily have. And I did not want to believe that was possible. And Ashton, my wife, was like, we got to get him tested. And so we jumped through the hoops to get him to a nephrologist. And even our nephrologist was like, oh, no, I think it's probably something else. It's probably like Bartter syndrome, or maybe it's diabetes insipidus. But we finally got him tested, and he did, in fact, have cystinosis, which is a really tough diagnosis to get. All of you guys know that. But this is Sam a little after being diagnosed. We had to get him in for a gastrostomy tube so that he could stay hydrated, get the good nutrition, and also so we could give him his meds. So this was a pretty tough hospital stay for us, but you can see Sam still trying to have a smile on his face. But after he got that G-tube, things really did start getting better. Once he was on medicines, started him on Cystigon, got him on all of his electrolytes, got things stable. It slowly started to get easier. But man, we were hooked up to that feeding tube pump all the time. It went everywhere with us. And Sam didn't really eat food at that point because it made him, throw up every time he put something in his mouth that wasn't water. So really I wondered, is this kid ever gonna eat food, or are we just gonna do this feeding pump forever? About the time that Sam was a little older than 2, we thought, we'd really like Sam to have a sibling. And we thought, maybe it's time to think about having another kid. And we figured, if you're gambling, 75% chance that he'll be healthy is pretty good. So let's roll the dice. And so we went ahead and We had another kid. This is Sam holding Lars, little Winston Churchill cheeks. And we got Lars tested right away because we knew there was like a pretty decent chance he could have cystinosis, and he also tested positive for cystinosis. So we got him started on treatment right away, and he's done pretty well, thankfully. So this is them now. I guess this is actually a few years ago. I need to update this slide, but it's been really great having them grow up together, and they have, even though cystinosis, it's a really hard disease, they have that in common, and they are really supportive of each other. Here's a picture a couple years ago at the Day of Hope. Right after Sam was diagnosed, we met the Sturgis family, so he's really good friends with Henry Sturgis. Both Sam and Lars really look up to Henry. They're sitting up here on the front row with him. But Sam was really excited to show Henry that he got his gastrostomy tube removed, and so they were showing off their scars. So it took a long time, though, to get to a point where he could, take all of his meds by mouth and not require, the G-tube anymore for medications, nutrition, but we're really proud of Sam for getting there. The main reason we come to Day of Hope is so that our kids can be with your children and to feel, like they're part of this community where people get what they're going through. And after last year's Day of Hope, as we were driving home to Utah, we thought, man, 3 days is just never enough. We need a longer Day of Hope. We need like a summer camp, And so that's when we started talking. Maybe we should do like a camp cystinosis. And so, we asked a few families that we had met at Day of Hope if they would feel comfortable sending their children to Salt Lake City. And so we did this last August, or was it? I don't even remember now. August or July, last summer. And we had a blast. You can see we had eight teenagers, or I guess Lars is technically not a teenager, even if he acts like one. But we had eight kids with cystinosis come out, including Brooke's mom, Monique, to help out. And honestly, we could not have survived without Monique, so thank you so much for coming. We kind of bit off more than we could chew. It turns out 17-year-olds are challenging, but we had a lot of fun. They came out. We took them up to Heber Valley, up in Utah, up in Midway, and we took them down the Provo River. We had a lot of fun staying up late every night, playing board games and eating charcuteries. Like, I've never seen kids eat so much salami in my life. And they had a lot of fun. They bonded. This is in the Midway Crater, which is like this volcanic hot spring that you can go swim in inside a mountain. And they just immediately started doing synchronized swimming together. It was really, really incredible just how bonded they were. So if you're interested in sending your kid to a cystinosis summer camp, let me know. We're thinking about maybe doing one again this August. So Ben Alexander, I've recruited to help me wrangle the teenagers. So if that's something that you think your adolescent might be interested in, please let me know. All right, let's get to business now. So we're talking about the foundations of cystinosis, and so really you have to understand a little bit of cellular biology, right? So the human body is made up of 40 trillion cells. And each of those cells has all these different compartments in them, right? So there's the Golgi apparatus, there's this endoplasmic reticulum and ribosomes, but really the most important compartment is the nucleus, because that's where the genetic material is, right? So that's where your DNA is, and that DNA is this double helix that's tightly wound up into these X-shaped things called chromosomes. And they really do look like X's if you look at them under a really powerful microscope. So this is showing a karyotype, which is an individual's chromosomes. You see you have 23 pairs of chromosomes. Half of them are from your mom, half of them are from your dad. And so what is DNA exactly? I would say that DNA is a library of instructions for how to build different proteins. And so one segment of DNA that gives you the instructions for one protein is called a gene. So when you hear a gene, that's instructions for building a specific protein. And your cell has special enzymes that can actually read this double strand of DNA and copy it into a transcript called RNA. And then that RNA is fed through special machinery called a ribosome to actually produce a protein that's made up of these building blocks called amino acids. So you'll see up here that this protein has, this chain of amino acids. We've got tyrosine, serine, glycine, another serine, right? And it's these bases that tell the ribosome which amino acid to attach to the chain. So, when we were trying to figure out how to explain this to our kids, my wife came up with this brilliant analogy that it's a lot like Legos, right? So these are your instructions for building a race car, and so that would be analogous to the DNA and the RNA. And then you have this master builder like Emmett from the Lego movie, who is your ribosome. So he's the guy who's actually going to build the protein. And then these are the actual building blocks that you need to build that race car, right? So these are going to be your amino acids. And the finished product is a protein. In this case, it's a cool race car with a spoiler. So then what happens if you have instructions for building a protein, but part of the instructions are missing, right? So that last step there that tells you to put the spoiler on, now it's gone. So we got a mutation there. And that's what a mutation is. It's basically any disruption in the instructions. And so this is exactly what happens in a genetic disease like cystinosis. You're missing part of the instructions so that when the ribosome goes to build the protein, it built a race car, but it doesn't have the spoiler on it, right? So that race car could probably still drive, but it just doesn't look very cool. So there's lots of different types of mutations that you can have, right? So these are the different bases that you have in your DNA, and sometimes the machinery pops in an extra one and it shifts everything down, right? Because the ribosome is reading these 3 at a time, and so if you add an extra base, then it's going to shift everything. Or sometimes it's missing one, and so that will also shift it. Sometimes it's missing more than one base. Sometimes it's missing 57,000 bases. Right, so remember each person has 23 pairs of chromosomes, and back in 1998, Dr. Corinne Antignac in France actually discovered where the cystinosis mutation was, and it's on the 17th chromosome right here. It turns out that Dr. Stephanie Cherky was working in Dr. Antignac's lab at the time and helped with this. Right, so here again is that 17th chromosome, and this is where the mutation for cystinosis is. There is a gene on the chromosome right here that codes for a protein called cystinosin. Wonder why they named it that. So there's been a lot of different mutations identified, over 100 over the years. By far the most common one is called the 57KB, which stands for 57,000 base pairs. So that's a really big chunk of DNA that was deleted at some point. And about anywhere from 50 to 75% of people of Northern European descent will have a copy of that gene. Some people have 2 copies of that gene. Some people don't have that gene at all. And then there's all sorts of other mutations that you can have. And cystinosis is an autosomal recessive disorder, which means that you need 2 copies of the gene, the mutated gene, to have the disease. And if you only have 1 copy, you're considered a carrier. And if 2 carriers get together, then anytime they have a kid, there's a 25% chance that kid will be unaffected, right? They'll get 1 good copy from mom and 1 normal copy from dad. They might get 1 normal copy from mom and 1 abnormal copy from dad, and that child would be a carrier. Or they might get an abnormal copy from mom and be a carrier. Or they might get 1 abnormal from mom and 1 from dad, and then that child would have cystinosis. So you see that anytime they have a child, there's a 25% chance that they're going to get that combination where they have 2 copies. And you'll notice that these are both mutated, but they're different mutations. So you don't have to have the exact same mutation as the one from both parents. So this is a picture of the cystinosin protein from the New England Journal of Medicine, and just showing that there's so many different places that you can have mutations in this protein. And also you'll see that this protein is embedded in a membrane of something called a lysosome. So lysosome, is one of those compartments in the cell that we were talking about. And in this picture, the lysosome is this thing that kind of looks like I guess it looks like a chocolate chip cookie or maybe a pizza. And the lysosome's job is to be the recycling center in the cell. So it's there to break stuff down so that the cell can use those amino acids again to build new proteins. So if there's like an old protein, or say the cell like gobbles up like some bacteria and it needs to chew that bacteria up, that's going to happen inside lysosomes. So here's my really terrible rendition of a lysosome, this orange circle here. But say we have a different race car now it's an old race car, it's broken down, we need to chop it up so that we can build new race cars. So we have enzymes in that lysosome that their whole job is just to chop up proteins. So in this case, it's Lloyd, the green ninja. But he's going to chop up that protein into all these different amino acids now, right? So we have all these building blocks. And each of those building blocks gets out of the lysosome to go be used again through a different transporter, right? So, that one goes out through the gray transporter, that one's going out through the white transporter, and that one's going out through the blue. So that's exactly what happens with the amino acid cysteine. It actually has its own transporter protein called cystinosis that it gets out of the lysosome through when it's been broken down. But if you have a mutation in the DNA or the instructions to build that protein, then that protein isn't going to work. So you can't get the cysteine out. So when the cysteine tries to exit the lysosome, can't get out, and it's just going to build up inside that lysosome. It accumulates and it crystallizes, and it'll kill the cell. And that's happening in every single cell in the body, all 40 trillion cells, right? And so that's why you see so many different systemic manifestations of cystinosis, from the eyes and the cornea to the kidneys, which we're going to hear more about today from our nephrologists. But it affects the muscles, it affects the bones, it affects the endocrine organs. Every cell is affected by cystinosis. And so, fortunately, in 1976, there was a researcher named Dr. Jerry Schneider at UCSD that discovered a drug called cysteamine that actually will go into the lysosome, and it will bind to the cysteine, and it creates a new molecule. And that molecule is actually able to exit the lysosome through a different transporter protein that's not cystinosin. And so they're able to actually get the cysteine out of the lysosome. So we call that cysteine depletion therapy. And cysteamine, the first iteration of that was Cystigon, approved by the FDA in 1994. Many of you have been on that medication or are still on that medication. And it works really well at getting rid of cysteine. So it's a really miraculous drug, and we're very fortunate to have cysteamine. Cystigon's tough. It's every 6 hours, and definitely has a lot of side effects: nausea, abdominal pain, the halitosis with the sulfurous odor. In 2013, the FDA approved Procysbi, which is a 12-hour formulation, which I think makes adherence a lot easier. You don't have to wake up in the middle of the night to take it. You still are going to have a lot of those side effects, but maybe they're delayed just because it takes longer to be absorbed. And as you all know, it's a very expensive medication. So, looking at our insurance claims, I kind of totaled up. It's about $112 a pill in the United States. That's an estimate. So for my son Samuel, he's on 12 pills twice a day. So, that's $2,700 a day, $80,000 a month, close to $1 million a year for that medication. And we know that cysteamine is not a cure. It's very good at depleting cysteine, but the disease marches on, and it doesn't fix everything. And so that's where gene therapy comes in. Because cystinosis is a genetic condition and affects your DNA, you need to actually fix the DNA to fix the disease. And so that's where gene therapy comes in. And Dr. Cherokee has developed this treatment with lentiviral gene therapy through an autologous stem cell transplant, where you take a cute little kid with cystinosis and you take some of their hematopoietic stem cells, You use a virus to put the corrected copy of the gene back in, and then you give those stem cells back to the person with cystinosis. Now, when I first heard about this, I was in medical school. I thought I knew a lot, and I was like, that doesn't make any sense. Why would fixing somebody's blood cells fix cystinosis? Cystinosis affects all the cells in the body. It affects the kidneys, the muscles. So if I give someone an autologous hematopoietic stem cell transplant, wouldn't that only fix the blood cells, right? So you see here, these are the hematopoietic stem cells. They come from the bone marrow, and they can turn into any kind of blood cell. So they are pretty remarkable cells. But why would that fix someone with cystinosis? It didn't make any sense to me. So it wasn't until I came to Day of Hope and heard Dr. Cherokee talk that things started to click. But what happens here is you have these stem cells turn into monocytes. It's a very specific type of blood cells. And those monocytes then differentiate into something called a macrophage. Now, macrophages are really cool cells because they are kind of the cleanup crew in your body. So if you have like an infection, you have white blood cells that go in and kill bacteria and viruses, and they make a big mess when they do it because they're just dumping toxins and just exploding everything. And so the macrophages come in to clean up the mess. And so because they're there to clean up the mess, macrophages have a ton of lysosomes in them, right? So these little pizzas are lysosomes inside the macrophage. So this macrophage is coming in and gobbling up the remains of some bacteria, and then it's going to fuse with these lysosomes, and the lysosomes are going to chew it all up and digest it so that the cell can use those things again. So because macrophages have so many lysosomes, they're kind of like these really expert lysosome delivery trucks. And so here you have like a muscle cell. This is a proximal tubule cell in the kidney. This is a neuron, right? So all of these cells have sick lysosomes in them if you have cystinosis. And what this macrophage does is it actually sends out this tunneling nanotubule that Dr. Cherokee has discovered and described, and it's going to send a healthy lysosome into that cell, and it's going to take out the sick lysosome. So it really is like an Amazon delivery truck just full of lysosomes, right? And these macrophages are just going all over the body, and it's looking for sick cells and saying, here's some healthy lysosomes, right? So this is how the stem cell treatment works, which I think is pretty incredible. There's a lot of other things that the CRF is studying, and Nancy talked about some of these. Some of the things I'm most excited about are new versions of cysteamine, other cysteine depletion therapies that maybe don't have the same kind of side effects, that maybe don't have the same kind of dosing schedules. I'm pretty excited about potentials there. New treatments for eye disease. There's a lot of research going on with inflammation, autophagy, nutrient sensing, that has yielded a lot of potential therapeutic targets. And so those are still things that are being studied in development. So I just want to say thank you for your attention, and thank you to the CRF for everything you're doing.
Thank you, Dr. Jenkins. Does anyone have any questions? Hi. Okay, great.
I was just curious, when the cysteine links with the cysteamine, and it exits through the lysosome, is it the cell able to use the cysteine that's exited? Like, what happens to the cysteamine once that separates inside the cell?
Yeah, that's a great question, and I don't understand all the biochemistry that happens after that, but yeah, that cysteine then can be metabolized and used. Yeah, it's a great question.
Any other questions? Okay,
All right, thanks.
thank you so much.